Thanks, Helen. Okay, so I'll just introduce myself. I'm from the Department of Primary Industries and Rural Development in New South Wales, and I've been working, in fact, I grew up on a blueberry farm and then I circled back to blueberries about 10 years ago in my research. So I'd like to first of all thank the organisers for giving me the opportunity to talk today. It's always a pleasure to talk about our research. And the work I'm talking about comes about because of the Clean Coastal Catchments project, which is a New South Wales government funded project, part of a larger initiative to try and reduce the risk of nitrogen pollution running off into off our coastline and damaging our marine environment. And part of the strategy included this project and a focus on several industries, and one of the industries selected to work with was is the blueberry industry, where you, I'm sure, are all familiar. Off the Coffs Coast of New South Wales is a very large area, or the largest area of blueberry production in Australia. So with that in mind, we formed a very large team, and I might add, including some very generous growers to work with. And I have to do a little shout out here to Diana and Leanne, who are our technicians who work with the crop, who maintain the crops on and off our research facility and collect most of the data. So I wouldn't be here without them. Okay, so back in 2019 we started this work, and our challenge was first of all to try and understand what the issues were that we were dealing with. So we worked with 4 growers. So we had four blueberry farms. They were all growing the variety 1111, which is a southern high bush variety, growing as an evergreen. And 3 of the crops were in soil and 1 of the crops was in substrate. And what we did was we monitored the soils and the plants for 2 years. We looked at their chemical composition. So we did sampling across that time. We looked in the soils also below the substrate crop. And what we found was that most of the soils were highly acidic, and they had high availabilities of aluminium, which is potentially toxic to plant growth, and low availabilities of calcium and magnesium, and in fact low enough that the plants had deficient levels of calcium and magnesium in their leaves, and the leaves also showed high values of nitrogen and phosphorus above those optimal levels. So the conclusion was that the practice or the practices were overusing or heavily using nitrogenous-based fertilisers. And our opportunities then were to look at how we might be able to reduce in the substrate crops, the nitrogen in the drainage, because that can be captured and then treated in different ways, but also at the input, so at the point of application of nitrogen to the plants. And so that's what I'm going to talk about in this session. So with that in mind, we established a tunnel facility growing blueberries in substrate at different levels of nitrogen, 5 different levels of nitrogen. And this is a more recent picture of the crop, just after we did a full harvest last year. And I've put a circle around the drippers and fertigation pipes there, just to give me a signal that I need to tell you that all of the nutrition, so we had a complete nutrient recipe for each of our 5 treatments with nitrogen varying between 40 and 120 milligrams per litre nitrogen, or parts per million. And that was, our target EC was 0.9 decisiemens per metre, so that's the strength of the solution, the concentration, and the pH was 5.7. And we have a whole environmental control system that adjusts the solutions according to the weather. And we've been monitoring that crop since 2021 when it was first planted. So it's almost 4 years old now, which is incredible to think about. Okay. So in terms of the experimental design, when you do plant nutrition experiments with your element of interest, so in our case nitrogen, what you aim to do is to grow the plants at different levels of your nutrient and hope to capture in that experiment plants that take up not enough nitrogen through to plants that take up potentially toxic amounts of nitrogen in our case. So what we did, after 1 year that the plants had been in the greenhouse, so 1 year after they'd established, the plants looked like this. And for each of our nitrogen supply treatments, we killed 3 plants per treatment. So we pulled them out of the pots, we cut them up, we dried them, and the dry weight of the whole plant is a good estimate that we use for growth. And then we also measured the nitrogen concentrations in those plants. And so this is a diagram explaining what we found that 1 year after we planted that crop. So we had 3 plants that we fully destroyed growing at 40 milligrams per litre nitrogen, and they incidentally had 40 grams of total dry weight, so that includes the shoots and the roots. And they had very low nitrogen concentrations compared to all the other plants that we grew in this experiment. And then our next highest treatment, 60 milligrams per litre nitrogen, had 3 plants that were quite different. One of them grew higher than any other plant in the experiment. It was 95 grams, and it had a slightly higher concentration than the plants growing at 40. But 2 other plants were smaller. They had 60 and 65 grams of growth. But their nitrogen concentrations were higher. And so what this tells me is that within that bunch of plants, we've got some growing deficiently and some growing to nitrogen surplus. They've taken up too much. So in this case, in our system here that we've used, I would make a guess that 50 milligrams per litre was probably our optimum at that point in time. So that's 1 year into this crop. Okay, track to 2024. So last year was our first, 2 years later, last year was our first full harvest of this crop. And you can see Diana with her bag of blueberries on the left. And Leanne on the right, she's got a device called a leaf area index analyzer, and it essentially measures, it's a non-destructive measure of the canopy growth. It's a really fantastic way to monitor growth over time, which we do regularly. And so these are the results from Leanne's data. So what we have is the full canopy area as we increase the nitrogen supply treatments. And what we've got there is the values for each column. We have 3 plants, the data for 3 plants represented. And the little skinny bar at the top gives you an idea of how variable those data are. So the wider the bar, the more variable the data, the bigger the difference between the smallest and the largest values. And so what this tells me is that as you increase the rate of nitrogen between 40 and 80, you have basically a linear increase. The plants grow a canopy, a larger canopy as their supply of nitrogen increases. However, as you increase nitrogen supply beyond that, you get no benefit in terms of that canopy size. So the million-dollar question is, how well did these plants yield? And we have a less, I guess, a more variable response than I would have liked as a scientist to see. But this is real data, so this is what we got. And I've got lots of ideas about these results and how we got them. I would love to talk to anyone who has an interest in this area later, but all I'm going to say is that our two highest yielding plants were from that 80 nitrogen per milligrams per litre treatment. And so at this point, in order to, so our focus as a group is to try and reduce excess nitrogen in as an input into these systems. So what I decided to do is use my working hypothesis is that we've got an optimal level between 40 and 80 milligrams per litre, but I suspect that beyond 100 we have surplus nitrogen to requirement. So I separated the data into those two groups. And so when we're looking at, this is the yield, sorry I've lost my axis label there. So this is the yield per plant, grams per litre for the whole season. So the season was picking every 2 weeks between early August and November. And what it shows us is that there is certainly no benefit from providing surplus nitrogen. At 100 and 120, you're getting no more yield. In fact, you're possibly getting less yield. This was a really interesting result. So the plants receiving the high level of nitrogen, what I'm now calling high, surplus to requirement, had much smaller fruit. And I think there's two reasons for this. The first is that some of the plants in this group were growth depressed. They had small canopies and they produced low values of total yield, and the fruit was smaller also. So I think that's a fairly toxic response to the amount of nitrogen that they were receiving. Other plants had yield that was high, but they had small fruit. And I've seen it in a few of my experiments over the years, a response of high flower numbers, and I've got the flower number data for these for this experiment, but we haven't processed that data yet. So one of my ideas is that with the high levels of nitrogen are initiating flower bud, and the resultant excess flowers are just not supported by the growth of those plants. Okay, so this is what I did at the end of the harvest last year was to measure the nitrogen in the mature growth of those plants. And when you have nutrients that are mobile in the plant, so nitrogen, phosphorus, potassium, magnesium, what the plant tends to do is if that element is in excess, they go and park it in their older growth. And so if you compare that tissue, the nitrogen in those parts with other tissues, then you get an idea of how much excess nitrogen in this case is there. And so the plants receiving surplus nitrogen definitely had higher levels of nitrogen in those leaves. Okay, so I must be running to time. Okay, so my conclusions are that when we surveyed our crops at the beginning of this work, we showed that there was excess nitrogen in those systems, which was associated with poor soils, so acidic soils and limited nutrition in terms of calcium and magnesium. The other thing that I've concluded, and it seems obvious when I state it, but it's important to say that small canopies require less nitrogen in supply. So establishing plants need less than mature plants, but also we've been doing some work with different pruning strategies, light pruning versus hard pruning, and we're seeing very clear differences in the nitrogen needs. So that's interesting work that we'll be talking about at some point in the future. And I do believe that we have, in the systems that we're growing with substrate, we have excess nitrogen, surplus to requirement, and that is associated with poor production. So you've got potentially reduced yields, definitely smaller fruit, and perhaps plants whose longevity is limited. Okay, so I think I'll end on that note because I'm happy to take questions at this point.
Yep, and we do have plenty of time for some questions, if anyone has any. Is there any WA people? I know they had questions.
Some insight in, because you held the same EC level of 0.9. So you take out a lot of nitrogen, but you replace it with something else. You leave something else out. How do the other elements look like in your experiment?
Yeah, so I don't have the exact values here, but we did, as you increase nitrogen, obviously we reduced other elements. We tried to keep them within a range that was sensible, but that is obviously a limitation. What I will say is that the more nitrogen, and I didn't mention that our nitrogen sources, so we supplied that total nitrogen as a half ammonium and half nitrate. And so as we increased that nitrogen level, of course, you're limiting the total amounts of other nutrients that you can add in. So what we might be seeing is also that confounded, we call it. We can't separate that treatment from those other elements that are changing, but we might be seeing deficiencies of those other elements. So that's data that we haven't looked at yet. Does that help answer your question? Partly. Yeah, I definitely have the numbers and I'm happy to share them, yeah.
Was there a reason why you chose UAN, or I assume UAN if it's a 50-50 nitrate and...
No, our recipes used ammonium nitrate.
Okay.
Yeah.
Right. So was there a reason why you chose that form of nitrogen? And secondly, when you were measuring your drain, were you allowing for the conversion of your ammoniacal nitrogen to nitrate, and given the mobility of that through the substrate, when you were measuring your drain to account for losses out of the system, did you have you got a comment on that?
Okay, so we didn't adjust the recipes at all. We're looking at the conversion of ammonium to nitrate in the substrate, so we are doing some measurements on that. We adjusted our ECs according to the weather conditions.
Was there a particular reason that you chose 50-50?
Oh, was there a reason we chose 50-50? It was, I guess, it was based on what we found out other people used in the industry, and it wasn't as high as some users, but some users use 50/50. So we kind of went with the best bet, and I had previously grown plants at different ammonium to nitrate ratios. So we kind of just went with the best bet of 50/50. That was, you know, experimentally it was easy for us to do that, but it's something that we could look at this point going forward, changing those ratios to see what we do, especially if there's interest in us doing that. Really, come and talk to me. I'd really like to improve what we're doing so it's as commercially relevant as possible.
Did you monitor pH, and do you think this would be something that would be important for the blueberry root zone and how nitrogen would affect that?
Yes, we did monitor pH. We tried to keep it within that range of around 5.7, but I can't talk about I don't have off the top of my head the management specifics of how we managed pH to those levels, but we kept it within the acidic range. And yes, that will affect the conversion of ammonium to nitrate and nitrification and ammonification, but generally was acidic. Yeah.